Have you ever pondered the power of the invisible? The power that resides within the very building blocks of life? We're talking about the intricate tangle of genetic material, the DNA, that defines who we are, how we look, and how we function. Every living entity carries within it an unseen world, a genetic blueprint that shapes life as we know it. But imagine if we could change this blueprint, manipulate it to our liking. The power to edit life's blueprint, that's genome editing for you. Our journey to decode life's blueprint has been a fascinating one, stretching over decades of relentless scientific pursuit. It all began in the mid-20th century, when James Watson and Francis Crick unraveled the double helix structure of DNA, the molecule where life's instructions are stored. This marked the dawn of a new era in biology. Yet it was only the first step on a long road. In the 70s, scientists discovered restriction enzymes, biological scissors, that could cut DNA at specific sites. This was the birth of recombinant DNA technology, a crucial foundation for our ability to edit genomes today. Fast forward to the 90s, and we saw the rise of zinc finger nucleases, or ZFNs, and talons in the early 2000s. These were proteins designed to cut DNA at specific locations, giving us more control over genome editing. But the real game changer arrived in the 2010s with the advent of CRISPR-Cas9, a system borrowed from bacteria that could be programmed to target any DNA sequence. This revolutionary tool, discovered by Emmanuelle Charpentier and Jennifer Doudna, has since transformed the field of genetics, enabling precise and efficient gene editing on an unprecedented scale. And thus we stand today on the shoulders of these scientific giants, wielding the power to rewrite life. Now that we have this power, it's time we understand its intricacies. Genome editing is a scientific technique allowing us to make specific changes to the DNA of a cell or organism. It's like using a molecular scissor, cutting and pasting genes to correct or introduce traits. The most widely used tool is CRISPR-Cas9, a revolutionary method allowing us to edit genes with unprecedented precision. But how does it work? Imagine a word processor, but for genes. You could find a sequence of DNA, cut it out, and replace it with a different sequence. That's CRISPR-Cas9 in a nutshell. This tool is derived from a natural defense mechanism found in bacteria, which uses CRISPR sequences to capture snippets of DNA from invading viruses, allowing them to recognize and cut these sequences in future attacks. Now, let's talk about applications. In medicine, genome editing has the potential to treat and even eradicate genetic disorders. By editing the genes of a patient's cells, we can potentially correct the genetic errors causing diseases like cystic fibrosis, sickle cell anemia, and certain types of cancer. In agriculture, genome editing could revolutionize the way we grow food. We could engineer crops to be more nutritious, resistant to pests and diseases, and tolerant to harsh weather conditions. This could help us feed a growing global population in a changing climate. But with this power comes important ethical considerations. How far should we go in editing the genes of future generations? Should we allow the creation of designer babies, choosing traits like intelligence or beauty? And what about the potential unforeseen consequences of making irreversible changes to our genetic makeup? As we delve deeper into the world of genome editing, we must remember to balance our desire for progress with our responsibility to ensure we're using this technology ethically and responsibly. As we hold this potent tool in our hands, we must tread carefully, for with great power comes great responsibility. Our journey doesn't end here. In fact, it has only just begun. Genome editing, once a concept in the realm of science fiction, is now a reality. Recent breakthroughs have taken us to places we could only dream of. In the last couple of years, we've witnessed the emergence of CRISPR-Cas9, a revolutionary tool enabling scientists to edit genes with unprecedented precision. Ongoing research is constantly refining this tool and expanding its applications. We've seen it used in the fight against inherited diseases, cancer, and even in the quest to eradicate malaria-carrying mosquitoes. But this is just the beginning. The field is brimming with potential. A world where genetic disorders become a thing of the past is not too far away. A world where we can enhance our capabilities beyond our biological limits is within our reach. As we venture forth into this brave new world of genome editing, the possibilities are as vast as our imagination. Our exploration of the genetic frontier is a journey, not a destination. As we delve deeper, the marvels of genome editing unfold, revealing endless possibilities. Stay curious, stay informed, and remember the power of the unseen. Don't forget to like, share, and subscribe for more exciting journeys into the world of science.